I'm going to demonstrate Lab 2A, where I will use restriction enzymes to digest a plasmid. Plasmids are small, circular pieces of DNA. Restriction enzymes can cut a plasmid at specific locations, creating 2 pieces of DNA. I can later use those pieces to confirm that the genes of interest are present in the plasmid. Here are the reagents and equipment that I will be using. First, I have a tube with the plasmid, pARA-R or R, a tube with a mix of 2 restriction enzymes, BAMH1 and Hindi3, a tube containing 2.5x buffer, and a tube with distilled water. First, I will combine the plasmid with restriction enzymes and buffer and put them in a centrifuge to make sure they're mixed. Then, I'll put them in a warm water bath where they will digest for 1 to 2 hours. I will also do a control setup where I will combine the plasmid with buffer, but instead of restriction enzymes, I will use distilled water. To set up the experiment, I need to label my tubes. R+ is for the experimental setup, and R- is for the control setup. Now I'm ready to add the reagents. First, I'll add 4 microliters of buffer and 4 microliters of the plasmid to each tube. Then I'll add 2 microliters of restriction enzymes to the experimental setup, R+. Finally, I'll add 2 microliters of distilled water to the control setup, R-. Now I'm ready to centrifuge the tubes. Notice I'm evenly spacing the tubes in the centrifuge to make sure it's balanced. I'll run the centrifuge for a few seconds to make sure everything is mixed and at the bottom of the tubes. Now I'll transfer the tubes to a floating rack and then into the warm water bath. Only the bottom of the tubes where the reagents are need to be under the water level. The plasmids will digest in the warm water bath for 1 to 2 hours. I can leave these samples at room temperature, but if I am not moving on to the next lab right away, I can store them in a refrigerator or a freezer. In the next lab, I will run a gel with the samples to make sure that the RFP gene is present.